Hello, Cedric, do you hear me? Because we cannot hear you at all since the beginning. Try to speak with the microphone, maybe. We hear Ralf, but you.
Cedric, perhaps you have been muted by the moderators. Can you unmute?
We still do not hear you, Cedric. That's better. Go ahead, speak. Still not. Maybe Christian, maybe you can take the...
Yes, Cedric, I was about to suggest that I move on with my introduction, and maybe by the end we have it fixed. So hello, everyone. Good morning from Vancouver, and good evening in France and Europe, I guess. So my name is Christian Steidl. I'm a scientist at the BC Cancer Research Institute in Vancouver, Canada. And I'm also professor at the University of British Columbia, working in the lymphoma field. And it's my great pleasure to introduce today our distinguished speaker, Professor Dr. Ralf Küppers, for this Redtalks series on the pathogenesis of Hodgkin lymphoma. So Dr. Küppers performed his Ph.D. studies in the group of Klaus Rajewsky, and graduated on the topic of classical Hodgkin lymphoma biology, where he developed methods for the isolation of Hodgkin Reed-Sternberg cells, and showed a clonal derivation of these cells from germinal center B cells. And in my view, and in the view of many, these studies are still to this day absolutely groundbreaking and cannot be missed in any pathogenesis model of Hodgkin lymphoma. In 1995, then he started his own research group, and in the early 2000, he also did a six-month sabbatical at Columbia University, working with Dr. Riccardo Dalla-Favera. And this led to many fruitful collaborations and exchange of ideas in the field of germinal center biology. And now for almost 20 years, Ralf is full professor of molecular genetics at the Institute of Cell Biology in cancer research at the University of Duisburg-Essen in Germany. And as you can imagine from his impressive track record and productivity and the training really from the best mentors in the field, Dr. Küppers represents a very wide field of work, and that involves the development and differentiation of B lymphocytes in physiology in humans, but also in cancer. And obviously his main disease focus to this day remains in the pathogenesis of both B and T cell lymphomas, and in particular Hodgkin lymphoma, as you will hear today. And as a Hodgkin lymphoma researcher myself, I'm personally very excited to hear Ralf talk today. And he will surely describe the current understanding of the pathogenesis of Hodgkin lymphoma, and lead through genetic lesions of malignant cells, tumor microenvironment interactions. But I also heard that he might also talk about nodular lymphocyte predominant Hodgkin lymphoma. And without further ado, I'm happy to now welcome Dr. Küppers to the stage. And Ralf, please take it over. Very excited to have you today.
Yeah, thanks a lot. Okay, so if Cedric still doesn't work, then I think I simply start with my presentation. I think it's easier to have questions in the end, as we discussed before, because I think I will not see the chat while I'm speaking. And so then I think I'll try whether this works out to share my screen with you. Okay, so Christian, can you confirm that you see the full screen of my presentation?
Yeah, perfect. Video and audio is nice.
Okay, yeah. So thanks a lot, Christian, for the nice words you had for the introduction, and also thank you to the organizers for inviting me to give a presentation in this series. I enjoy this. It's a nice initiative to have this online seminars on lymphoma biology over the borders in a way. Yeah. So as you heard, my topic today is pathogenesis of Hodgkin lymphoma. Just start with one brief introductory slide. I think most of you will know that. But yeah, to set the stage. So as you certainly know, the tumor cells are called Hodgkin and Reed-Sternberg cells. I will mostly abbreviate them as HRS cells in the following in classical Hodgkin lymphoma. But as you already heard, I will also briefly touch on the rarer subtype of Hodgkin lymphoma, nodular lymphocyte predominant Hodgkin lymphoma, which is quite distinct. And here the tumor cells are called lymphocyte predominant or LP cells. So that's that you know that. And a common theme of both types of Hodgkin lymphoma is that the tumor cells, so the HRS and the LP cells, usually represent in the range of 1 or less than 1% of the cells in the tumor tissue. Here you can perhaps see in the lower right side a typical examples. I think that's from a frozen tissue section. Here you can recognize two multinucleated or binucleated Reed-Sternberg cells, but most of all the remaining cells here, which are not stained for CD30, are microenvironment cells. So you could say you have about 99% microenvironment in this lymphoma. But this, of course, has for a long time and still hampers the molecular analysis of this lymphoma. Because you need to typically have approaches like microdissection or cell sorting if you really want to work with pure or enriched Reed-Sternberg cells. Now, what is relevant for today, of course, also is the origin of these cells, and work from our group, but later also from other groups, I think clearly revealed that HRS cells likely derive from what we termed crippled germinal center B cells. So germinal center B cells, which were engaged in T-cell dependent immune responses, but which acquired in the germinal center so-called destructive or crippling mutations, so that most likely many of these cases cannot express anymore a B-cell receptor. And indeed, perhaps related to that, actually HRS cells have largely lost their B-cell gene expression program, their B-cell phenotype, which was also quite confusing to identify the origin of these cells. An important aspect for the presentation today is that HRS cells are practically always CD30 positive, as is seen here also on this example staining down there, important marker for the diagnosis of this lymphoma, but also perhaps for the biology, as I will allude on. And whereas HRS cells somehow are like failed germinal center B cells, non-functional B cells, it seems that the LP cells of nodular lymphocyte predominant Hodgkin lymphoma more resemble in their gene expression pattern and in the structure of their B cell receptor genes transformed but positively selected germinal center B cells, which still express the B cell receptor immunoglobulin on their surface. Now, I selected 4 topics for today. I hope I can do this nicely in 45 minutes without having to rush too much. So as already briefly mentioned by Christian, one of the topics will be to give you, let's say, a state of the art or state of the current knowledge about genetic lesions in HRS cells. So this is more like a summary of published work by many groups. And then I have 2 parts of my presentation which are somehow centered around CD30. On the one hand, what are normal CD30 positive B cells and what is their relationship to the CD30 positive HRS cells? This is a published study. But then I will present a still unpublished work where we are still doing the final experiments. And this is a project about the role of CD30 on the HRS cells or in the HRS cells. And then finally, I would also likely raise your attention to a study that was also, I think, published 2 years ago, but which I find remarkable and some of you might not have seen, and that is on the role of the B-cell receptor specificity in nodular lymphocyte predominant Hodgkin lymphoma. So these are the 4 topics I would like to address with you now. And so let's begin with the first one. So some remarks or an overview about our current knowledge about genetic lesions in HRS cells. So as an introductory slide here, So the work on that, I think, really started in 1999. So after microdissection approaches to isolate HRS cells from tissue sections have been established first by us, and then later by several other groups. I think 1999, that was then the first paper on IKB alpha mutations in HRS cells. And then it started to look for point mutations in isolated HRS cells. We had then in the last 20 years a number of papers from different groups on that. And this was more recently very much extended in a way by the first exome sequencing studies of isolated HRS cells. Most of you might know these studies. Reichel et al. Blood 2015, Enrico Tiacci also in Blood 2018, and Wienand et al. In Blood Advances 2019. Right now, there is also, I think, the first whole genome sequencing study early online in blood cancer discovery, not yet mentioned here. And then for, let's say, thinking about the landscape of genetic mutations in HRS cells, it's also worthwhile to mention, of course, that also recently the first studies on circulating tumor DNA in Hodgkin lymphoma patients, I listed here the first three most relevant papers, were also published, so that we now, let's say, have a number of papers available, which give us, I think, a first good insight into the landscape of mutations in Hodgkin cells. And funnily, in a way, the whole thing started, I would say, with the identification of the IKB alpha mutations. And indeed, the NF-κB pathway turned out to be of major relevance for the pathogenesis of classical Hodgkin lymphoma. So here, first of all, I show you a typical cartoon of the classical NF-κB pathway on the left side and the alternative NF-κB pathway on the right side. Of course, I will not go into detail here, but you know there are surface receptors like members of the TNF receptor superfamily members, CD30, CD40, which signal through the NF-κB pathway, which activates then kinases, which can inactivate the inhibitors like IKB alpha, which I already mentioned. And then once they are destroyed by proteasomal degradation, then in the canonical pathway, heterodimers or homodimers of NF-κB factors can translocate in the nucleus and then regulate the expression of hundreds of typical target genes. And when it was recognized first with Hodgkin cell lines and then on tissue sections that practically all cases of classic Hodgkin lymphoma show strong constitutive NF-κB activity in the HRS cells, then one after the other genes was analyzed, and this now gives the current state on that. So as already mentioned, NF-κB alpha or NF-κB inhibitor epsilon mutations are recurrent events in Hodgkin lymphoma, like affecting 10 to 20% of the cases. But then later also we and others showed several years later that a further inhibitor of the NF-κB pathway A20 or the gene name TNF-AIP3 is actually even more recurrently mutated, like 40% of the cases overall. Also a main inhibitor of the canonical pathway. Then it's well known from cytogenetic work that one of the NF-κB factors, REL, shows gains or amplifications of the gene in about 40% of the cases. And similarly, I think without going all into detail, also mutations were found in the non-canonical pathway, like Nick Janes. I think one of the contributions here, important contributions from our chair, Christian Steidl. We reported rare TREF3 mutations, also tumor suppressor gene in this pathway. And perhaps finally to mention here, as we know, about 40% of the Hodgkin lymphoma cases are infected by the Epstein-Barr virus, and in these cases, the latent membrane protein 1 of EBV is expressed, which can actually activate strongly both the canonical and the non-canonical NF-κB pathway. And overall, it seems, that a large fraction of Hodgkin cases shows mutations in one or perhaps even several of those genes, so that there is a strong genetic contribution to this constitutive NF-κB activity. And here already, I think the motto is not a specific gene is needed, but apparently Hodgkin Reed-Sternberg cells need a strong and constitutive activity of NF-κB for their survival and malignant growth. And a multitude of components of this pathway can contribute to this activation. So this is the first, let's say, major story of recurrently mutated genes in Hodgkin lymphoma. Some of the most frequently mutated genes can be assigned to the NF-κB pathway. We have a similar situation in another major signaling pathway of lymphocytes, and that's the well-known JAK-STAT signaling pathway. As you certainly know, this is the main signaling pathway for cytokines. So if cytokines bind to cytokine receptors, then this activates JAK kinases, which are bound to these receptors. These kinases then phosphorylate themselves and the receptor and STAT factors. And once the STAT factors are phosphorylated, they can homoheterodimerize, translocate into the nucleus, and then again function as transcription factors and regulate the dozens or hundreds of genes. So a major transient signaling pathway also in normal lymphocytes of the immune system, but also here multiple mutations in different genes were found. I think the first finding were inactivating mutations in the SOX1 gene, a major negative regulator of JAK-STAT signaling. This is mutated in 40 to 50% of the cases, so one of the most frequently mutated gene in HRS cells described so far. But later also PTPN1 was identified, another negative regulator to be recurrently mutated. Here also Christian contributed or was, I think, the first to publish that. Then also based on cytogenetic data with FISH analysis, gains of the JAK2 kinases were also found quite frequently in 30% of the cases. And more recently, one of the STAT factors, STAT6 mainly, was identified to have gains of the gene locus or activating point mutations together in about 30% of the cases, and more rarely also STAT3 or STAT5b mutations were found. So again, here the motto in a way is apparently Hodgkin cells need essentially a strong and constitutive activation of the JAK-STAT cytokine signaling pathway for their survival. But perhaps besides cytokine signaling itself, so physiological signaling, genetic lesions play a major role in the deregulated and constitutive activation of this pathway. Just as a side remark, when Hodgkin cells have a gain of JAK2, this can also have epigenetic effects because the JAK kinase is known that it can also modify the histone H3 and thereby have an influence of the epigenetic regulation, but this only as a side remark. So these are two main fields of genetic lesions. A third field links to the microenvironment, or to be more specific, links to immune evasion. So this is now, let's say, not a specific signaling pathway, but more a general field. But also here, I would simply like to give a review about the current knowledge. So because you could argue in particular in the EBV positive cases of Hodgkin lymphoma, why are these Hodgkin cells which express viral antigens not recognized by cytotoxic T cells, for example? Or why are they not eliminated by NK cells when they downregulate MHC class 1? So apparently the Hodgkin cells manage to evade from and control by the immune system. Many factors are contributing to that, and I will not address this today. But what I want to allude here is genetic events which contribute to immune evasion, and I think the four perhaps most relevant ones are listed here. So first reported by Reichel et al. In the exome sequencing study, perhaps it's not 50% of the cases, one would say nowadays, because later studies found lower frequencies, but still it is clear that a good fraction of classical Hodgkin lymphoma cases have inactivating mutations in the beta-2 microglobulin gene, which is essential for MHC class 1 expression. So that would be a way how the Hodgkin cells could evade from an attack by cytotoxic CD8 positive T cells, because they need MHC class 1 on their target cells. But then on the other hand, if the Hodgkin cells are MHC class 1 negative, they could be principally recognized by natural killer cells, which are specialized in eliminating MHC class 1 negative cells. And here, one, or at least in a fraction of cases, a factor why NK cells are inefficient, besides other reasons, could be that at least in some cases of Hodgkin lymphoma, there are mutations or deletions of the CD58 gene, and this is a surface protein which is needed for full activity of NK cells on their target cells. Then here on the lower left side, again, important contribution by Christian Steidl to that work, because he was the first to report about translocations affecting the MHC class 2 transactivator and thereby impairing MHC class 2 expression also in a considerable subset of cases, like about 15%. And that would be a way how the Hodgkin cells could evade a detection by CD4 T cells, which can partly also be cytotoxic in some conditions. And then finally, perhaps in terms of recurrency, the most important here, and also clinically, perhaps very important, is the finding by several groups, including Green et al. In 2010, that actually many cases of Hodgkin lymphoma have gains or amplifications of the PD-ligand 1 and PD-ligand 2 genes, so these checkpoint molecules. And these are molecules then which inhibit the activity, as you certainly know, of CD8 positive T cells, perhaps also additionally of some macrophages and NK cells. So that is a further important likely way how HRS cells evade by genetic means from an elimination by cytotoxic cells in the microenvironment. So an important, I think, field of that there are numerous mutations which somehow help the Hodgkin cells to be rescued from an attack by the immune system. But of course, besides those three groups, now the recent also the exome sequencing studies and targeted sequencing studies revealed a number of additional recurrently mutated genes. This here is just, I think, a selection of further genes which are, let's say, in the range of 10% or more percent of the cases show this. So we have also, let's say, some classical mutations like in the tumor, well-known famous tumor suppressor gene TP53 or the FUS, the CD95 receptor. I think interesting is also the quite frequent mutations in the exportin-1 gene, XPO1, which regulates nuclear export of RNA and proteins nearly in a quarter of the cases. Point mutations nearly always in the same codon of the gene. But then also there is an emerging picture that also the PI3 kinase AKT pathway, which is also well known to be constitutive active in nearly all cases of Hodgkin lymphoma, and this is not only mediated through signaling through surface receptors, but that also here mutations in genes like GNA13 or ITPKB could contribute to this constitutive AKT activity in the HRS cells, which is also clearly a pro-survival factor as functional studies in cell line showed. And finally, there is also something on epigenetic regulation or chromatin structure. So there is no indication, although the frequencies differ very much between different papers, of mutations in histone genes themselves. So to summarize this more, let's say, general overview, I would like to make the following points and perhaps also some let's say, idea for discussion later. So there are indeed many mutations in the NF-κB and JAK-STAT pathway among the most recurrently mutated gene. Here it is clear that apparently it is important that these pathways are always active in the Hodgkin cells, but it can be different genes that mediate this in combination, most likely also with signaling through this pathway. Then there is an important role of immune evasion for Hodgkin lymphoma pathogenesis. I showed you, let's say, the key examples of that, which are also quite recurrent events. There are many further mutations which now have been identified by these exome sequencing studies or also by some targeted sequencing studies by our group and other groups, but those are mostly at rather low frequency, so are quite heterogeneous. And perhaps one remarkable point is that actually nearly all of the mutations, with few exceptions that have been found so far in Hodgkin cells, are also known from other B-cell non-Hodgkin lymphomas. I mean, often it's very similar in primary mediastinal B-cell lymphomas, but there are also, let's say, in other mutations, you have mutations in the JAK-STAT or NF-κB pathway, for example, and some are curiously, I think when we started, we and others started to look for that, there was perhaps the hope to find genetic reasons for this loss of the B-cell phenotype, like this reprogramming of the Hodgkin cells. But in my opinion or my understanding, there are so far no mutations identified which would, let's say, clearly explain the lost B-cell phenotype, so like activating mutations in some master regulators of some other immune cells were so far not really frequently found or that could explain this. Therefore, to bring this together, why is Hodgkin lymphoma, the classical Hodgkin lymphoma, so very different in the appearance, in the phenotype of the tumor cells, but having nearly the same or let's say related set of mutated genes? perhaps it's on the one hand the combination of genes, like this combination of NF-kappa B and JAK-STAT pathway, that in that combination is not seen in many other B-cell non-Hodgkin lymphomas, that, let's say, defines the specificity of Hodgkin cells. But I would like to speculate that it's perhaps also the specific origin of the Hodgkin cells, so that it makes a difference, let's say, whether you get a JAK-STAT pathway mutation in a positively selected germinal center B cell or in a post-germinal center B cell, as opposed to the crippled cell, which has already a different setup. But I think this is purely a speculation, but perhaps I might be interested to hear your thoughts about that. Okay, so this was the overview I wanted to give about genetic lesions. So therefore, I would now like to turn to the second topic I would like to address. So that is our previous work on normal CD30 positive B cells and their relationship. So as I said in the introduction, Hodgkin cells are always CD30 positive, and therefore I think it was an obvious question to ask, okay, is this just an activation marker on these cells, or is this somehow pointing perhaps to a relationship to normal cells or to the origin of these cells? Because CD30 is also considered as a activation marker on normal B cells and T cells, although only in some situations, and it's expressed by rare cells. So here, and taken from a paper from another group from 2005, you can see in blue, in a lymph node or in a tonsil, here a few CD30 positive cells on the one hand outside of germinal centers, so extrafollicular, but also in the germinal center. You see a few of those cells, just less than 1% of the cells. Some of them are T cells, many of them are B cells, and therefore we wondered, okay, if you think Hodgkin cells come from germinal center B cells or developed in a, let's say, post-germinal center part of phenotype, can we perhaps better understand this? And the question that we have raised in that study was, okay, what about the normal CD30 positive B cells? What are these cells in the germinal center? Are they normal parts of the germinal center reaction? What is their V gene setup, and what is their gene expression pattern, and what is the relationship of these normal CD30 cells in the gene expression pattern to HRS cells? And similarly, what about the normal CD30 positive B cells outside the germinal center? There are hardly any studies about that in the literature. So what is the cellular origin of these cells? Are they pre-germinal center? Are they post-germinal center? And again, are they a distinct subset, or are these just cells with a particular activation marker? And that's why Mark Weniger in the group, or beginning first Enrico Tiacci and later taken over by Mark Weniger, or finished by Mark Weniger in the group, we isolated these cells from human tonsils and then performed, at that time, still Affymetrix gene expression profiles of these cells, compared them to normal B cells and to other lymphoma cells to better understand what are these normal CD30 positive B cells in the human. And this now here shows a first result of that. I will address only some data of this paper, and that was an unsupervised hierarchical clustering, where we compared here on the left side the CD30 positive germinal center and CD30 positive extrafollicular B cells. So we separately sorted them from the tonsils by FACS, and then made separate gene expression profiles from 5 samples each. And then we compared them to conventional germinal center B cells, memory B cells, plasma cells, and naive B cells. And what you can recognize here is there is a dramatic upregulation of hundreds of genes in the CD30 positive B cells, even compared to the highly active conventional germinal center B cells. So apparently, normal CD30 positive B cells are somehow like super activated cells, upregulated of hundreds of genes. But indeed, there is a clear distinction between those two types of germinal center B cells, like the germinal center ones and the CD30 extrafollicular ones. And it's also clear here now, CD30 is not simply a further activation marker, for example, on germinal center B cells, because hundreds of genes are strongly upregulated in comparison to the germinal center B cells. So then, of course, the question about what were the differences? What is this upregulation? What is the key feature of these cells? And here, since the data are already published a few years ago, I just focus on 1 or 2 points. And perhaps the main exciting thing for us in that finding was when we performed gene set enrichment analysis. You might know these typical pictures from gene set enrichment analysis, that we recognize that both types of CD30 positive B cells, so both it's the germinal center ones as well as the extrafollicular non-germinal center ones, showed a massive activity of MYC target genes or upregulation of MYC target genes. So apparently normal CD30 positive B cells have a very high MYC activity. We then also validated by Western blot that indeed the cells, or by QRT-PCR and then even by Western blot, that indeed the normal CD30 positive B cells have the highest transcript levels of MYC in comparison to the other B cell lineage subsets we looked at. And they indeed also, we could confirm protein expression by Western blot in these cells, whereas bulk germinal center B cells or memory B cells do not express the MYC protein. So that is clearly a peculiar finding of these cells, besides also, let's say, for example, NF-kappa B signature. But here I focus on this finding that these are MYC active B cells. And this was particularly interesting for the germinal center B cells, because here we could relate our findings to earlier very elegant work by the Dalla-Favera and Klaus Rajewsky groups on a mouse model, where they wanted to understand the role of MYC in murine germinal center B cells, because those papers now 11 years ago or 10 years ago revealed that MYC plays a central role in the initial phase of the germinal center reaction, and then is important to sustain the reaction. So that apparently these mouse data indicated that MYC is expressed by a small subset of centrocytes, so positively selected germinal center B cells in the so-called light zone of the germinal center, and that these are the cells which are instructed to return to the dark zone to become proliferating centroblasts again, and then further proliferate and undergo hypermutation without MYC expression as centroblasts. And so that let's say MYC, NF-kappa B drives centrocytes to become either memory B cells or plasma cells, but MYC might be the main regulator for the return or for the decision that centrocytes become proliferating centroblasts again. And we now think that by our work we have identified the human counterpart of these cells, because we have seen now that it is the CD30 positive germinal center B cells which are high in MYC activity. So we think that these are the cells which are prepared, or which are preparing for return to the dark zone for further proliferation, and that this is the key feature of these normal cells. Now, of course, then the second part of this work was to compare the normal CD30 positive B cells to the lymphoma samples that we had. And this, I show also here only a selection of the data that we generated. And this is now a principal component analysis, which on a two-dimensional scale compares the similarity of our samples. So each of the circles represents one sample with one Affrimatrix chip. And here we compared conventional germinal center B cells in black with CD30 positive extrafollicular B cells in blue. And then we put in, for this comparison, how do follicular lymphomas, diffuse large B cell lymphomas, and HRS cells of classical Hodgkin lymphoma behave for that. And as you can see here, follicular lymphoma, as to be expected as the prototypical germinal center B cell lymphoma, cluster close to conventional germinal center B cells. DLBCL in yellow are quite heterogeneous, also more as expected. But then interestingly, our, I think, 16 samples of classical Hodgkin lymphoma in green here cluster close to the normal CD30 positive B cells. So indeed, there is apparently a substantial overlap in the gene expression between the normal CD30 positive B cells and the HRS cells. So it's not only that they share CD30 as an activation marker, but that they have more similarities. And this is also, let's say, partly described in the paper. Just to mention, NF-kappa B activity and MYC target genes are some of these common features of normal CD30 positive B cells and the HRS cells. But then the question is, okay, but what is the difference? Like, say, are the normal cells already half Hodgkin cells in that way? But here, let's say, focusing on main difference between the normal CD30 cells and the Hodgkin cells. I just have here a summary here. As here you can see, the paper was published in 2018 in the Journal of Clinical Investigations. So normal CD30 positive B cells still express all the B cell markers, so they do not have this lost B cell phenotype, which is a hallmark of Hodgkin cells. They have even a stronger signature of strong proliferation, like MYC and E2F target genes, than the Hodgkin cells. I think, which is quite remarkable, considering that Hodgkin cells are also proliferating tumor cells, but MYC activity is even stronger in the normal cells. And on the other hand, looking at what is higher in the HRS cells, then typical features of HRS cells, like regulators to remodel the extracellular matrix, like metalloproteinases, MMPs, are higher in the Hodgkin cells, and this hallmark of Hodgkin cells, that they secrete tons of chemokines and cytokines. This is also much more strongly seen in the HRS cells than in the normal CD30 positive B cells. And finally, also the well-known expression of non-B cell genes in HRS cells, which contributes to their strange phenotype, like CD3 components, granzyme B or ID2. That is only seen in the Hodgkin cells, not in the normal cells. So therefore, we concluded that key features of HRS cells are indeed still disease-associated and not shared with the normal CD30 positive B cells. So that is published work that we did, where we wanted to understand the relationship between the normal CD30 positive B cells and the HRS cells. But then more recently, we became also interested to understand the role of CD30 itself now. Why are Hodgkin cells always expressing CD30? And is this just reflecting their activation phenotype, or is this indeed of pathobiological relevance? And this is now a part which is not yet published, which I would like to share with you. So here I think I need two introductory slides. So just to remind you, CD30 is a member of the TNF receptor superfamily. The official name is TNF receptor superfamily member 8. It's mainly expressed by some rare activated B and T cells. And as I already told you in the previous part, normal CD30 positive B cells are found in the germinal center and in extrafollicular regions, but they are typically very rare. CD30 is not only expressed in classical Hodgkin lymphoma, as most of you certainly know. It's also a hallmark of the T cell lymphoma, anaplastic large cell lymphoma, and of the B cell lymphoma, primary mediastinal B cell lymphoma, as well as in some other B cell non-Hodgkin lymphomas, like in some further cases of diffuse large B cell lymphomas and in primary effusion lymphomas. It is not only relevant for the differential diagnosis of lymphomas, but it is now also of highly relevance for these type of CD30 positive lymphomas as a therapeutic target, because at least the hematologist among you certainly know that there is this brentuximab vedotin, so a drug conjugated anti-CD30 antibody, which is now used in some patients with CD30 positive lymphomas to attack and to kill the lymphoma cells. And I think this even makes it even more relevant to understand, is CD30 expression just an activation marker, or is there more about that? Now, what is known about CD30 signaling? I mean, of course, something is known as the TNF receptor superfamily. So CD30 signals after ligand binding as a trimer. It's a membrane, of course, surface membrane molecule. It has no own enzymatic activity. So signaling is mediated through TNF receptor associated factors, abbreviated TREFs. And CD30 typically can bind TREF1, 2, 3, and 5, according to the literature. And it is well known that a main signaling pathway downstream of CD30 is the NF-kappa B pathway, as indicated here briefly, but there is also indication for other pathways which might be activated upon CD30 triggering, and there are data in the literature, at least for the MAP kinase ERK pathway as a downstream signaling pathway in CD30 positive cells. Now, of course, when it was identified that there is CD30 expression as a hallmark of HRS cells, people became interested already, let's say, many years ago. Is there signaling? What could happen? Then a few points on that. So indeed, it is the case that in the Hodgkin lymphoma tissue, you find CD30 ligand expressing cells, which could trigger CD30 on the Hodgkin cells. These are mainly eosinophils and mast cells. they're often not in direct, let's say, proximity to the Hodgkin cells, but of course in the dynamics of the tissue, or perhaps through some, let's say, other factors like membrane vesicles, extracellular vesicles. One could imagine that there could be a triggering of CD30 through CD30 ligand positive cells. However, it has also been speculated in the literature that because of the very high expression in most cases of Hodgkin lymphoma, that there could be ligand-independent signaling even without direct binding of CD30 ligand in the tissue. But then, on the other hand, one could ask, well, is there need for CD30 signaling, for example, to activate NF-kappa B in light of the many other ways to activate NF-kappa B? And I gave you in detail an overview about the many genetic lesions in the NF-kappa B pathway. And I think I already mentioned perhaps that also other TNF receptor family members are expressed by HSs like CD40, which will also activate the NF-kappa B pathway when binding to CD40 ligand expressed by CD4 T cells. So one could argue, well, perhaps in the established Hodgkin clone, CD30 is not relevant anymore because NF-kappa B is in any way activated in these cells. And indeed, when several groups, mainly a group from Germany, Michael Hummel from Berlin, or the group from Japan, Watanabe et al., worked on this in like over many years, as you can see, starting in 2000. I think here are the four main papers. They worked with Hodgkin lymphoma cell lines, of course, to do some functional work. You have to stick to classical Hodgkin lymphoma cell lines, and they performed either stimulation studies or the down-modulated CD30 expression using shRNAs, but those studies gave discrepant results. So the Berlin group mainly reported there is no CD30 signaling in the Hodgkin cells, whereas the group from Japan concluded from their work there is, at least in some Hodgkin line, there is CD30 signaling. So that motivated us to also work on that. And now we used the CRISPR-Cas approach, because you could argue that with shRNA, perhaps the down modulation of CD30 was not strong enough. So therefore, Annika in our group, Annika Killian, in her PhD thesis, performed work where she now wanted to and successfully did knock out the CD30 gene in several Hodgkin lymphoma cell line to see if you have definitely eliminated CD30. Does this have an influence on the physiology, the survival of the cells? And I show you here one example for one of the Hodgkin cell lines, HTLM2. Annika used a control guide RNA and two CD30 specific guide RNAs listed down here. Guide 2 and guide 5 were the most efficient ones. You can see here in this fax picture, after knocking it down in the red, you see the staining for CD30 in the cell line treated with a guide RNA. And you can see that first you see still a staining. Actually, sorry, the gray, in here you see the down modulation of CD30 expression after 2 days and 6 days of culture, but those negative cells vanished after 30 days. And this is an important finding. So I have to explain you this picture down here. So when we knocked down CD30 in the control cells, then nothing happened because it was only a control guide RNA. The cells remain CD30 positive and remain surviving. So here the survival is measured. Whereas in those aliquots where we knocked down CD30 by guide 2 or guide 5 against CD30, we see that a few days after knockout, indeed, the surface expression of CD30 goes down. So we see this efficient knockout of CD30, so that the fraction of remaining CD30 positive B cells went down to, let's say, a few percent or 30 or 20%. But now, importantly, in these cultures, you have a mixture of HTLM2 cells with knockout of CD30 and other cells in which the Cas9 experiment was not successful. And after prolonged culturing, as you can see here, you see that the negative cells were overgrown again by the remaining CD30 positive cells, because at the end of the culture, as you can see here, most of the cells were again positive. And let's say this was not a reversion of the knockout, that is not possible. And we tested this also, but that was an overgrowth. So apparently CD30 knockout cells have a growth disadvantage in comparison to the HTLM2 cells, which remained CD30 positive, and the similar results were seen also in 3 other Hodgkin lymphoma cell lines. So indeed, this was the first indication or a strong point that CD30, even in the Hodgkin lines, where there's definitely no CD30 ligand around in the culture, that even in the steady state situation, CD30 is important for the optimal survival and proliferation of these cells. But then the question was, and we made a control with another gene, I do not show this here, that this is not a side effect of the Cas9 or an off target of CD30 guide RNA. But then the question was, why do they have a growth advantage? Is this perhaps apoptosis? A natural question for this. So now in this set, you see that we performed experiments where we measured annexin 5 as a measure for apoptotic cells on these cells by flow cytometry by FACS, looking after various days after electroporation as listed in the x-axis. And now the data here are normalized to the guide control. So one means identical annexin 5 positive cells like in the negative control. So always if the values are above 1, that means you have increased fraction of apoptotic cells. And indeed, as you can see here with these stars in 3 of the 4 Hodgkin cell lines, which we analyzed, HTLM2, UHO1, and KMH2, we saw a statistically significant increase cell deaths of the Hodgkin lymphoma cells upon elimination of the CD30 gene. We then looked for active caspase-3 as a let's say specific marker for real apoptosis, so not general cell death, but specifically apoptosis. And here, 2 of the cell lines showed statistically significant increase of active caspase-3. So there is at least, let's say, a substantial contribution of cell death. So apparently, knockout of CD30 induces cell death in at least 3 of the 4 Hodgkin lines that we analyzed as a main factor for this growth disadvantage of these cells. But then, of course, the question arose, okay, apparently knockout of CD30 has an effect on the physiology of CD30 cells. So what is this effect? And I think a natural way how to address this nowadays is to perform RNA sequencing to see whether knockout of CD30 influences the gene expression of Hodgkin lymphoma cell line cells. And this Annika did with 3 Hodgkin lymphoma cell lines as listed here. Again, we made the RNA-seq with a control guide RNA and with 2 different guide RNAs against CD30. And then, then we performed with these cells RNA sequencing. And then we focused on differentially expressed genes. We were quite stringent. So we only considered genes which were affected by both guide RNAs. So perhaps again to rule out potential off-target effects. And also we looked, let's say, for statistically significant up or down regulated genes. And just the numbers are here is not so important, but what you can see is that in the cell lines, several hundred to more than 1,000 genes were statistically significant, differentially regulated when we knock out CD30 in these cell lines. We sorted the knockout cells. Some were upregulated, some were downregulated. So then, of course, the question was which genes are up and downregulated. Again, we started off with gene set enrichment analysis to look for pathways which are regulated. And of course, the first thing to look at was the NF-kappa-B pathway, because I mentioned in the introduction that NF-kappa-B signaling is the best known downstream effect of CD30 signaling. But this was, you could say, in a way, a little bit disappointing for us because only 1 of the 3 cell lines for which we did this RNA sequencing showed a significant downregulation of NF-kappa-B activity by gene set enrichment analysis in A, or by pathway analysis in B, or then also in a functional study with an NF-kappa-B reporter and a luciferase activity assay to confirm this. So indeed, in HTLM2 cells, there is a significant downregulation of NF-kappa-B activity when we knock out CD30. So in this cell line, the presence of CD30 contributes to the strong NF-kappa-B activity. But in the 2 other cell lines, there was a tendency, but this did not reach statistical significance. So perhaps in these cell lines, the multiple genetic lesions in the NF-kappa-B pathway are so strong already that CD30 does not further contribute to NF-kappa-B activity in the, L42H and KMH2 cells. But when we looked at specifically differentially regulated genes, we were somewhat also surprised or excited to see that in this among the genes which were significantly down or upregulated, we identified a number of chemokines and cytokines. Most of them were downregulated. So everything below this line is again normalized to guide control. So everything below this line, which are 0.2, means downregulation. Up means upregulation. So several of the key chemokines, which are very well known. So CCL17 is TARC, one of the most highly expressed genes overall in Hodgkin cells, which attracts CD4 positive helper cells and regulatory T cells into the microenvironment, is apparently partly regulated by CD30. And we validated these findings by QRT-PCR. We are planning to do some also studies on IL-13 with ELISA, but this is still pending. So apparently several key chemokines and cytokines are in their expression co-regulated by CD30, which was, I think, so far not known or not obvious, that CD30 has this role to upregulate chemokine expression of several key chemokines in the HS cells. And finally, for this experiment, I come back to another topic. I mean, I mentioned already MYC in normal CD30 cells and MYC in Hodgkin cells. And therefore, we were quite excited actually to see that when we performed gene set enrichment analysis, we saw in all 3 lines a significant downregulation of MYC target genes, also B-cell specific MYC target genes. So apparently and unexpectedly, for me at least, CD30 contributes to MYC activity. And this was even more, let's say, clear and more significant than the NF-κB regulation, which was seen, as I said, only in one of the lines. So all the 3 lines where we knocked out CD30 showed a substantial downregulation of MYC activity. Then, of course, we wanted to confirm that this is really also affecting MYC itself. So we performed a Western blot for MYC protein. Here 2 exemplary Western blots are shown, and you can see indeed comparing guide control, so our negative control versus 2 guides against CD30, we see here the clear downregulation of MYC protein. So if we knock out CD30, MYC protein goes down. And this now also explains why we see the loss of MYC target gene signatures in these cells. And this might also be part of the answer to this toxic effect of CD30 knockout, because in previous work from our group by Anna Lollis, published in Leukemia 2018, and also I think partly reported by Lou Stout in another paper several years before, we already knew by shRNA that knockout of MYC is toxic for Hodgkin lymphoma cell lines. So therefore, it could be that the downregulation of MYC upon CD30 knockout contributes to the toxic effect that CD30 knockout has. So these are, I think, are for us quite exciting finding, and again pointing somehow to an important role of MYC in the pathobiology of Hodgkin lymphoma. And in future experiments, of course, we want to understand why is MYC downstream of CD30, which signaling pathway is used for that, how does CD30 in Hodgkin cells regulate the MYC protein levels. This is so far not clear, and yeah, we want to identify this. So to summarize the third part of my presentation, the work by Annika showed that there is clearly a growth disadvantage of a complete CD30 knockout in Hodgkin lymphoma cell line cells. We actually saw this partly also in cell lines of anaplastic large cell lymphoma in primary mediastinal B-cell lymphoma, but I did not show you the results here. But just as a brief information, this growth disadvantage is at least partly mediated by apoptosis. And then regarding the gene expression analysis that we did, we revealed that there is an altered expression of NF-κB target genes, at least in some of the cell lines, but there was a consistent downregulation of MYC activity when we knock out CD30, and apparently CD30 also co-regulates chemokines and cytokine expression. So therefore, we conclude from this work that CD30 indeed is not an activation marker on Hodgkin cells, but it contributes to the pathophysiology of these lymphoma cells. And I think this perhaps is a good news for treating Hodgkin cells with rituximab vedotin, because this might indicate that it's not easy for the Hodgkin cells, for example, to downregulate CD30 when the cells are attacked by bradtroximab vedotin, because they might often still need CD30 expression for their optimal survival. Okay, so this brings me to the last part of the presentation. This is a work, I'm only a co-author on that. It's a mainly work by Lorenz Thurner from Hamburg and Sylvia Hartmann from Frankfurt, Main in Germany. But I contributed to that, and I really like that work, and I think some of you might have overlooked that. It's published in Nature Communications like 2 years ago, but I think it's quite interesting for the pathobiology of nodular lymphocyte predominant Hodgkin lymphoma. I will be brief here because this is also already published, as I just said. But yeah, a few or one introductory slide. So you might, of course, know here in this audience that there is a role of antigens in the pathogenesis of various B-cell non-Hodgkin lymphoma, so signaling through the B-cell receptor as a pathogenetic mechanism. Autoantigen specificity of the BCR has been reported in chronic lymphocytic leukemia, some diffuse large B-cell lymphomas, many mild lymphomas, and others. There are also a few instances where there is a suspicion that infectious agents might trigger the B-cell receptor, like hepatitis C virus, a few cases with Epstein-Barr virus, specificity of the BCR, and perhaps Helicobacter pylori in the famous gastric mild lymphomas. Now, on this background, it's important or interesting to note that there is a consistent expression of the BCR in the LP cells of nodular lymphocyte predominant Hodgkin lymphoma, so a clear difference to classical Hodgkin lymphoma, as I mentioned in the introduction. And we and also other groups reported already many years ago that the BCR of LP cells is practically always somatically mutated, shows also a low level of ongoing hypermutation, indicating or showing that indeed the LP cells are antigen-experienced germinal center B cells. So there could be the idea that the BCR plays a role. And also for the following of relevance is that somewhat peculiarly about 25 to 30% of the cases of nodular lymphocyte predominant Hodgkin lymphoma have been reported to express IgD, and because most B-cell lymphomas, also those which do not undergo class switching, are expressing only IgM with very low or no levels of IgD, so often IgD is downregulated, and the lymphoma B-cell non-Hodgkin lymphomas often express an only IgM or class switch receptors, but that was quite a peculiar finding already known since many years. Now, Lorenz and Sylvia became very much interested to look for a potential specificity of the receptor of LP cells against bacterial antigens. So what they did, they cloned, so they microdissected LP cells. That was the work of Sylvia in Frankfurt and her team. Then they expressed the B-cell receptor of something like 15 cases of LP Hodgkin lymphoma, expressed the B-cell receptor from these single cells, and then tested them against extracts of major bacterial strains. And then the exciting and important finding was that in this test, in this dot plot, several cases of nodular lymphocyte predominant Hodgkin lymphoma showed a positive signal of their BCR for Moraxella cataralis. This is a bacterium which often can infect the throat of people. It's not life-threatening, but can make infections. And interestingly, all of the cases which showed Moraxella specificity, as you can see in this small line, were among those IgD-expressing LP cases or LP cells. So the BCR of 6 of 8 NLPHL cases with IgD-positive LP cells recognized extracts of Moraxella cataralis. And as a test, also 45 B-cell receptors of other B-cell non-Hodgkin lymphomas were also tested on these arrays, and not a single one showed specificity for Moraxella, which also excludes, I think, some technical artifacts or that. Then, of course, the next question was, okay, but what is the bacterial protein? What in these extracts is recognized? So different Moraxella cataralis proteins were tested by Lorenz Thurner in Hamburg. And then the interesting finding was that actually all of the 6 cases which showed specificity for Moraxella showed the same antigen that was recognized by the BCR, and that is the gene or the protein RPOC, which is a subunit of the bacterial RNA polymerase. This was confirmed both by Western blot, and which is shown here, and by ELISA. So apparently there is one common antigen which is frequently expressed by the surface B cell receptor of LP cells. And then the question was, okay, but if this is true, were those individuals indeed infected by Moraxella? So can we identify typical IgG antibodies against Moraxella in the serum of LP patients? So Lorenz and Silvia looked at nearly 100 cases of patients from nodular lymphocyte Hodgkin lymphoma and compared also nearly 100 cases of classical Hodgkin lymphoma from the German Hodgkin study group, and then performed ELISA for anti-RPOC antibodies. And I think the striking finding here was that you had only a few cases of classical Hodgkin lymphoma with very low titers of Moraxella, whereas 20 of 98 cases of NLPHL showed high titers of antibodies against RPOC, which somehow indicated that indeed many of the patients were infected by Moraxella and perhaps also repeatedly infected because of these very high titers. So that would fit with the idea that perhaps recurrent or long-lasting infections contributed to the malignant transformation of the LP cells or of normal germinal center B cells to become LP cells. And for the scenario that is developed on that, a further point, and that is the interesting finding that Moraxella cataralis also expresses a type of a superantigen. It expresses MIT-HAC, which is known since many years as an IgD binding protein. And keep in mind that I said that all of the cases with Moraxella specificity were IgD positive. And based on that, now this is the last data slide that I have, the following hypothesis or scenario was developed. So apparently, if a person or a patient is infected by Moraxella cataralis, then small vesicles are produced from the bacterium, which express on the one hand this MIT-HAC protein, which binds to the constant part of IgD and can thereby function as a superantigen and trigger the receptor. But then also, if a B cell is specific for RPOC, then also these vesicles will bind to the normal antigen binding site of the B cell receptor. So in a way, if you consider that then such an RPOC specific B cell is driven in a germinal center reaction, you can get a very strong immune response with many plasma cells, which then produces IgG, which was detectable in the serum of many patients. But then you could also think this is a very strong and perhaps repeated stimulation of germinal center B cells, which then might increase the risk that these cells underwent malignant transformation in the germinal center by acquiring oncogene tumor suppressor gene mutations, and that this then can give rise to the IgD positive LP cells. And this now also might explain why these cases remain IgD positive and do not downregulate IgD or undergo class switching, because then you still have this trigger through MIT-HAC on the constant part, so that at least, let's say, in the early stage of the disease, perhaps not in the established lymphoma clone, you have this constitutive triggering or strong triggering of the BCR of the LP precursor cells, if these are the RPO specific cells. So, and I think an interesting scenario about a strong triggering by a bacterial antigen as a co-cause for nodular lymphocyte Hodgkin lymphoma. And I can mention here, I think this is allowed, that Lorenz and Silvia continued their work, and they now submitted a further study, which is just under revision with a journal, that they now also in a further set of LP cases, which are not IgD positive. So other cases, they found a further bacterial specificity. So at least, it's perhaps not 100% of the cases, but in a considerable fraction of cases of nodular lymphocyte Hodgkin lymphoma, the BCR shows specificity for particular bacterial antigens, which therefore play likely a role in the pathogenesis of this type of Hodgkin lymphoma. Yeah, and this paper was published in Nature Communications 2020. And as I said, the follow-up paper is currently under revision and will hopefully in a few months appear in a journal. this brings me to the end. So mainly thanks to Mark Weniger and Rico, who were the driving force for the CD30 work. Of course, this was also part of my long-term collaboration with Martin Hansmann and his team. And as I mentioned already, the part Annika as the driving person for CD30 activity in Hodgkin cells and the role in the middle part. And then the last part, which I mentioned, was mainly driven by Silvia Hartmann and Lorenz Turner, and also, of course, including Martin Hansmann. Yeah, and with this, I'm at the end. I stop.
All right.
Yeah,
Thank you, Ralf.
Freigabe. Yeah.
Can you hear me? Oh. First, thank you so much, Ralf, for this fascinating
You're
talk.
welcome.
So it was very. I'm sure the attendees are very happy to attend this talk. It was a very update overview.
I'm sorry, I think I was a little bit long, right? I spoke an hour instead of 45 minutes, but I hope that's still fine.
No problem. we are on time for some questions in the chat, and just briefly, I have to present myself. I have a problem. Okay. I have to present myself to avoid questions at the end. I'm a hematologist in Dijon. I'm Cedric Rossi in France, and I'm involved in the LISA group. And currently, I would like to say hello from Stanford, because I'm a postdoctoral researcher on Hodgkin in Stanford in Alizadeh lab, particularly in CT and Hodgkin lymphoma. So we have a lot of questions, and we can start on perhaps the field on the role of CD30. We have some questions about CD30 in the pathogenesis and the role to detail more, to have more detail on this. Just one question from Jean-Philippe Merliot. Talk about ask for, are there differences in CRISPR-Cas9 activation of CD30 in the different cell lines according to their genetic status? Could you give us some details about that, Ralf?
So you mean the efficiency of knockout, or I mean
Yeah,
for Solans that we used, yeah. So they are somewhat different. I mean, we used that, perhaps I haven't said specifically, we used an approach where we had preloaded Cas9. So we started with some lentiviral experiments, but they were not so efficient. So the experiment that we did was that we bought Cas9 protein, loaded it with the guide RNAs, and then performed nucleofection with the AMAXA system. That is quite reasonable. There are some good protocols where you get quite efficient transfection of the Hodgkin lines, but the four cell lines differed somewhat in the efficiency. Yeah, I focused here on, I think I showed mainly the pictures of HDLM2. I think that was the most efficient one, but also the others worked. And also then, perhaps not surprisingly, also the results were somewhat different. We did not yet really link that completely. I mean, we know from these cell lines, for example, which NF-κB pathway genes are mutated. So, for example, I made this point to say when we knock out CD30, there was a significant loss of NF-κB activity only in HDLM2, not in the other three ones. That was only a tendency that we still have to sort out when you write up the paper. But it's known that several of the lines have mutations in IκB alpha, epsilon, or A20. So that is our speculation. I mean, that then perhaps in the other lines, NF-κB is activated already by other means so strongly that their knockout of CD30 does not influence significantly the NF-κB activity.
And just in general about CD30, if we build a bridge between pathogenetic genetic lesions and therapeutic applications, can we move forward? Can you give us your feelings about how can we move forward to personalized medicine in Hodgkin lymphoma? For example, if we have CD30 pathway with mutations in κB, for example, can we treat patients with brentuximab vedotin, or is it unuseful? Or what is your feeling about that?
I mean, first of all, one has to keep in mind, of course, as you know, I'm not a clinician, so I'm not very much in the clinical studies. I try to read them, but of course, I do not treat Hodgkin patients myself. So of course, we have to keep in mind that brentuximab vedotin does not interfere with CD30 signaling. Here, the idea is the antibody binds to CD30, then the complex is internalized, and then in the cells, the toxin, a microtubule inhibitor, which is linked to brentuximab vedotin, or which is part of it, then kills the cell. So here, CD30 is only used as a cargo to bring in the immunotoxin into the cell. Now, looking at the pathway, of course, you could say, for example, with our finding that apparently a major effect of CD30 is MYC activation, then you could think to interfere with that. But as I think we all know, MYC itself is a difficult target. I think there are some clinical studies and a lot of companies try to find MYC inhibitors because that would be so relevant for many cancers. But as far as I know, this is not very much advanced. So whether, let's say, based on that work, one could think of a targeted therapy to interfere with MYC, because it was already previously known actually that MYC knockdown is toxic for Hodgkin cells. But other information, let's say what is in between, what might be more easy to target downstream of CD30 than MYC itself, that is something we do not yet know, and we might have to work on.
Thank you.
Okay, fantastic talk, Ralf. So I'm having a question from Bertrand regarding the Hodgkin lymphoma pathogenesis and the speculation that combined mutations might be leading to the specific Hodgkin lymphoma phenotype. Bertrand is challenging a little bit if that is really the only explanation, and could there be also a concept of, and I loosely translate here the question, so-called driver microenvironments and an external stimulus, at least in combination? And I'm reading into the question if you can speculate about the order of is there driver microenvironments first and then later mutations, or first mutations and then synergy? Do you have any insights or opinions about these two concepts?
That's a good point. I mean, my thinking still is that there has to be some genetic or viral thing in the beginning. So my thinking is coming from this crippled germinal center B cell, so that at least a quarter of the cases have clearly destructive mutations, and it's so well known that if a germinal center B cell acquires destructive mutations, it would immediately undergo apoptosis. So something must prevent the apoptosis of the Hodgkin cells, and there I see EBV as a major player. But also, let's say, mutations in the NF-kappa B pathway or JAK-STAT pathway, which then, let's say, at the first wave, I assume provide survival signals to keep this pre-apoptotic germinal center B cells alive. But then these cells survive, but they are not anymore functioning as normal cells if they have downregulated BCR expression. And then perhaps the cells try to find ways to survive by interacting with the microenvironment, by changing their phenotype. And there I fully agree also with the direction Bertrand and you point to. It could be that it's not, let's say, master mutation in a notch factor or something like that, which, let's say, imposes some T cell genes, but that could then be mediated epigenetically or through interactions with the microenvironment. So if the Hodgkin cells start to produce strange chemokines and cytokines and now closely interact with rosetting CD4 T cells, that this might contribute to changing the phenotype. I mean, for example, in the EBV cases, there is indication that LMP2 could contribute to downregulation, partly downregulation of some B cell genes. So it is clear it doesn't have to be oncogenes, which, let's say, impose this reprogramming of the cells. But perhaps people have thought or had hoped to find something as an easy explanation. But I fully agree. I mean, so far, also the exome sequencing papers, they do not really have this other master regulator, which could explain that. I mean, there is, let's say, this, just thinking about this from Stefan Mattas, the receptors, the mulet receptor CSF1R, that there you could think that mix produces some signaling which promotes some non-B cell genes. So there are some indications for a few of the oncogenes, but not, let's say, the super gene which we have all or many have been looking for. And yeah, and I agree, it could be some microenvironment influences, which then perhaps might influence only epigenetics of the cells. That leads to the downregulation, because we know that many B cell genes are down modulated also because of epigenetic silencing in Hodgkin cell lines, at least that has been shown. So yeah, it might mainly be a consequence of microenvironment interactions. I fully agree. Yeah.
Yep. Thanks for the insight.
And regarding EBV infection, we know, and in your talk, it's really in our mind to this role of EBV pathogenesis of Hodgkin, and how we have the conclusion and the overview, the perfect overview. We can ask for what is the role and participation of EBV across all Hodgkin lymphoma. For example, we know EBV is not prognostic in younger patients, but this virus has a prognostic unfavorable effect in older patients, Hodgkin lymphoma, over 45 years old. So, what is your feeling or your insights about the specific mutations across the age, perhaps regarding EBV virus on Hodgkin lymphoma, and what is the balance between EBV viral role and the mutational profile outside the EBV virus?
Yeah. Good questions, and I can only partly answer them. So I think it's now getting more evident that, or partly evident, that, let's say, indeed it could be complementary roles. So I pointed out that in the EBV positive cases, LMP1 is expressed, latent membrane protein 1, and this is a strong NF-κB activator. And indeed, there is now, I think, quite good evidence from our group and others that in general, let's say, those cases which are EBV positive, they have less frequently mutations in genes like IκB alpha or TNF ARP3. There we also reported that. So apparently, as I said, the Hodgkin cells need NF-κB. If they are not EBV infected, then they need the mutations in IκB alpha gene or TNF ARP3 gene as genetic drivers for NF-κB activity, whereas if by chance an EBV harboring B cell was driven into a germinal center reaction and in the germinal center expresses latency 2, including LMP1, then that will give a strong NF-κB signal, and those cells are less dependent on acquiring additional mutations. So there is now, I think, the indication that indeed EBV has a pathogenetic role. It's also important to mention here that regarding this really crippling, I mean, I try to be short, not to be too long. So the real crippling mutations are seen in about a quarter of the cases. So crippling mutations which prevent B cell receptor expression at all. Whereas in other cases, we think there are disadvantages, point mutations, so like low affinity mutations, which are also disadvantaged and would promote apoptosis, but they would still allow BCR expression. And now the interesting thing is, if one distinguishes those two types, that actually all of the cases where we identified crippling mutations were EBV positive. That we reported in a review actually only a few years after we collected already a number of cases. And therefore, there it's relevant that in the EBV positive cases, a further latent membrane protein of EBV is expressed that's called LMP2A, and this mimics a B cell receptor because it has a similar signaling as a B cell receptor. And therefore, there is a scenario that if a germinal center B cell acquires a crippling mutation which destroys BCR expression, like a premature stop codon or a frame shift, this cell can apparently only survive if it is EBV infected at that stage, because then LMP2A can kick in and mimic the presence of the B cell receptor, and only then the cell can survive long enough to give rise to a Hodgkin case. So that is even more, let's say, pointing to an important role of EBV, at least in those cases where the BCR is completely destroyed by somatic mutations. Now, the other aspect of the question, that there is, let's say, also age difference, like children in Central America are nearly 100% EBV positive if they have Hodgkin. I think children in Germany are relatively less frequent EBV positive, or the young adults, the nodular sclerosis cases are less. I think here I do not have a good answer to that. I mean, we have to keep in mind that nearly everybody in the world is EBV latently infected. It's typically in about 1 in 1 million of our B cells, mostly memory B cells. So the chance that an EBV positive B cell is driven into a germinal center reaction is low. But therefore, there is a possibility that some germinal center B cells get transformed without EBV and others with. And yeah, I think I do not have a good answer there. So why young adult Hodgkin cases are mostly EBV negative, whereas childhood cases from Central America or older mixed cellularity cases from Europe are mostly EBV positive. I do not have a good answer to that. It's an interesting phenomenon, but I'm not aware of any good explanation for that, unfortunately.
Okay, we have one more question here on the chat from Dr. Ritchie, and this relates to CD30 positivity and potential escape from CD30-directed treatment through antigen loss potentially. There's another dimension to that question. So first question is, how frequently do you think that can happen? And now, in the light of your data that shows that CD30 is important and has a connection to MYC, makes it interesting, because then an antigen loss would be selected against. Very interesting. But then the specific question from Dr. Ritchie is that this could be counteracted still. So MYC then down, and then with all the consequences on metabolism, so it's glycolysis is mentioned here, and could then be compensation mechanisms imaginable. And have you done any work into that direction, or is that something separate?
Yeah, thanks. Good question also. Second part to begin with, no, we haven't done anything, but of course, I mean, there might be escape mutations, or it might be that at later stages of the disease or in the microenvironment, where there are many factors functioning on the Hodgkin cell, CD30 is less relevant than we see in the Hodgkin lines. Of course, we have to be careful. We did this work only with Hodgkin lines, which are adopted to grow in culture for a long time. And under the pressure of treatment, there might, of course, be escape mutants, for example, which then acquire additional mutations, which upregulate NF-kappa B or MYC activity in these cells. And we did not do anything on metabolism so far. But of course, yeah, that's a good question. Regarding the first part, perhaps even I'm not completely up to the literature. So of course, that would be interesting to see what is happening in those patients which escape treatment by brentuximab vedotin. And actually, I do not know. I would have to do more literature work there. So whether people already looked at those patients, and then to see, for example, whether then CD30 is mutated, or how it could be that then or whether these cases which escaped actually downregulated CD30, and whether this was due to mutations, as we know in some other targeted therapies where the target mutates away from the treatment. But I actually I'm not aware that this has already been carefully analyzed by any study. But it would be, of course, be very interesting to look in clinical studies on those patients who escaped or who relapsed after brentuximab vedotin therapy, whether then the Hodgkin cells are CD30 negative or low. But perhaps I'm not completely up to date on the literature there. Yeah.
And just I have a short question about your very interesting graph on PCA analysis. The dots about DLBCL close to Hodgkin are from GCB DLBCL, I guess, and the APC are far, or is Is it correct or no?
Yeah, so the cases that we had, I mean, this was from a, let's say, we did not analyze these cases, I must admit, really, let's say in detail, whether they are more ABC-like or GCB-like or T-cell rich B-cell lymphoma-like. It was just cases that we got hold on without doing this work at that time. So of course we know, let's say partly, that's also I think partly work from Christian, that some subsets of DLBCL, like primary mediastinal B-cell lymphoma, are actually quite similar also in the overall gene expression pattern to classical Hodgkin lymphoma. So we have not done this detailed analysis of these few cases of DLBCL which we had on our Affymetrix chips. But I would also speculate that there might be a collection of different cases, and that, for example, the GCB DLBCL were more closely to the normal germinal center B cells, and perhaps the ABC likes, or if we would have a case of, but I think we didn't have a case of at least not that I know of, primary mediastinal B-cell lymphoma, that they would be more close to the CD30 positive B cells, which also, let's say, the strong NF-kappa B signature, which is also a hallmark of ABC DLBCL. But we did not, as our focus was on Hodgkin, I must admit that we did not look in detail on these, let's say, 6 or 8 DLBCL cases that we had there.
Thank you.
Yeah, and maybe mediastinal gray zone lymphomas are an interesting entity in that context. Yeah. So, so what, Cedric, maybe we allow one more question before we conclude. Does that sound reasonable?
Yes.
We have Bertrand asking a question very globally on what in vivo and ex vivo tools are available, because as presented today, and as I can also tell stories myself, most is cell line work. And do we have anything on the horizon that is better than that?
No, I think that I know that for many years, for example, also Klaus Rajewsky tried to make, let's say, a complex mouse model with EBV genes and others to generate a mouse model. But I mean, there was, let's say, 1 or 2 single papers, but to be honest, I think there is no mouse model of Hodgkin. I mean, of course, the only thing is that people used immunodeficient mice strains and simply transplanted Hodgkin lymphoma cell lines into them. But that, of course, is, I think, not what one means, because then you will not get a Hodgkin with a microenvironment, because these mice then lack normal B and T cells, because otherwise the Hodgkin cells would not grow. I know that several groups tried to grow primary material in the cells, like Ursula Kapp in Max group like 20 years ago. But there the problem was that once the normal T cells or the normal human cells die away in these immunodeficient mice, then also the Hodgkin cells die. So I think the primary Hodgkin cells need their microenvironment. And so if you transplant suspensions of Hodgkin lymphoma lymph nodes after a few let's say, a short time, nothing remains in the cell, so that also doesn't work. And there is no, let's say, genetic mouse model. I think it's so complex, and really, let's say, making a mouse model for pre-apoptotic germinal center B cells is quite a challenge to model that really. So there is nothing. And then second part of the question, and I see also do not something directly on the horizon, I must say. I'm not aware of any mouse model. I think, Christian, you also not. And then primary cells, I mean, we also, for other also genetic work, we try to sort them. So not only doing microdissection on tissue sections, but I think based on the seminal work of Jonathan Fromm, who established multicolor staining and approach for de-rosetting of rosetting T cells. Nice work from him. We also use this to enrich Hodgkin cells. But if you do this with a lymph node section, lymph node piece you end up with a few hundred cells, or perhaps 1,000 or 2,000 cells. So you can use this then for RNA sequencing, or perhaps for next generation sequencing. That's also something that's ongoing in our group, which I did not present today because it's too preliminary. But I think for functional work, let's say doing stimulation in vitro, you simply you do not get from the patients enough material. That's a pity. Yeah.
So thank you very much. I think we have to conclude. Thank you once again, Ralf, for the very wonderful talk. Thank you, Christian, for participating and your expertise. So it's very appreciated. And thank you, Juliette from Kalim, for organizing this kind of session. We have to repeat the Hodgkin session. Very, very important and very fascinating disease. So
thanks a
thank
lot
you,
to all the
everyone.
people listening or hearing and phrasing questions, and of course also to you as the chairperson. Thanks a lot. I enjoyed this. Yeah. Okay. Goodbye.
Thank you.
Goodbye.
Bye.